Assalamualaikum and a very good evening to all lecturers. Today, our group will be presenting about analysis of elastin.
Thank you, Zulfarhan. Here we talk about the introduction of elastin. But before I will talk about the introduction of elastin, I would like to tell you why we choose this protein. We choose this protein because of its simplicity in nature. Now talking about the elastin, elastin is in skin and tissue of the body. Elastin is insoluble in water. Initially synthesized as a precursor of polypeptide called tropoelastin. It consists of approximately 700 amino acids with molecular mass of 700 kDa. It is one of the most components of reticular dermis. Elastin is found in all animals. Next slide. The structure of elastin. It is a connective tissue of protein and rubber-like property. Elastin and glycoprotein microfill are present in lungs, walls of arteries, elastic ligaments. Looking at the picture, that is the work of elastin. Elastin is responsible for stretching and pinch in our body. The more we grow, the older we grow, the quicker the elastin weakens. Next stage. Function of elastin, as we all know, it plays role in protein and connective tissue that allows main tissue in the body to resume their shape after stretching or contracting, and also produced by skin that is responsible for resilience in elastic fiber. Next slide. To talk about the characteristics of elastin, elastin is a very long-lived protein with a half-life of about 70 to 78 years in human skin. It is more flexible than collagen that provides resilience and elasticity to tissue and organ. Elastin's amino acid sequence are glycine, valine, alanine, and proline. Here, I'm going to hand over to the next person to continue the source of elastin due to the time. Thank you.
Okay, thank you, Vera. So for the source of elastin, elastin is found predominantly in the walls of arteries, lungs, intestines, skins, and other elastic tissues. Next slide. So to get more efficient results in elastin purification, the tissue needs to be washed with saline to remove soluble protein and deflated. The mincing or grinding process steps also will be much more easier to get a fine powder in nitrogen liquid if the tissue are washed in the saline. Next. So for the purification part, there are 4 methods that can be used to purify elastin, which are first the hot alkali, or also known as Lansing method. Next, the autoclaving method, which also known as Petrich method. And the last are 2 mild isolation approach, which are Starcher and Demon method. Next. So from the 4 methods that we found, we think that the Starcher method is the most effective method to purify elastin. It is because due to harsh condition in hot alkali, protein was degraded throughout the process, and this must be considered while planning experiment with the pure product, while the autoclaving method is less harsh than using hot alkali, but the finished product will include more contaminant. So Starcher method is the most effective method because it combines autoclave treatment with extraction using reducing and chaotropic agents. Because elastin lacks methionine, cyanogen bromide is used to cleave non-elastin protein. So the product obtained using this technique can be equivalent to elastin prepared using hot alkali with less internal peptide bond degradation. Okay, thank you. Next, I will pass to Zul.
Thank you, Haziq. Now I will talk about the materials used in structure method. One of the material used is 0.05 mole disodium phosphate buffer with pH 7.6 containing 1% of natrium chloride and 0.1% EDTA. Next is 30 milliliter of 0.1 mole tris buffer pH 8.2 containing 0.02 mole calcium chloride. 4 milligram of trypsin and 200 milligrams of cyanogen bromide is used. Not only that, 30 ml of 0.05 mole tris buffer of pH 8 containing 6 mol of urea and beta mercaptoethanol is used. Lastly, 10 ml of 97% formic acid is used. Next, I will talk about the procedures in such a method. After the elastin tissues is washed, the tissues will be extracted. The pulverized tissue will be extracted for 72 hours with repeated changes of 0.05 mole of disodium phosphate buffer of pH 7.6 containing 1% natrium chloride and 0.1% EDTA. And then the residue will be washed twice with distilled water and lyophilized after the final reaction. And the lyophilized material will be suspended 200 milligrams of will be suspended in 200 milligrams of the lyophilized material in 30 ml of water and then autoclave for 45 minutes at 25 IB pressure. And then the sample will be centrifuged and washed and the residue will be washed twice with water and then suspended with 30 ml of 0.1 mole tris buffer of pH 8.2 containing 0.02 mole of calcium chloride. Then it will be incubated with 4 milligrams of trypsin at 37 degrees Celsius for 18 hours. And the sample will be centrifuged again and the residues will be washed with water and suspended in 10 ml of 97% formic acid. 200 milligrams of cyanogen bromide is added and the suspension is shaken in a well-ventilated fume hood at room temperature for 5 hours. And the sample will be centrifuged again and the residue will be washed twice with water and resuspended in 30 ml of 0.0 mole tris buffer of pH 8, which contains 6 mole urea and beta mercaptoethanol. Lastly, the suspension is stirred overnight at room temperature and which will then centrifuge, wash successively with 3 washes each of ethanol and acetone. And lastly, it will be dried in vacuum. Next, I will pass to Sri to explain the next part.
Thank you, Zul. For sequencing method used in Elastin is amino acid analysis, which is consists of several repetitive amino acid sequence, which is VPGVP, VPGVG, VPGVGV, and more on. So it is highly insoluble and extensively cross-linked. Matured link elastin is formed from tropoelastin. Tropoelastin is secreted from elastogenetic cells, which oxidation by lysis oxidase. Subsequent protein-protein association give rise to a massive macroarrays of elastin. The structure of tropoelastin consists of an alternative of hydrophobic regions responsibly for elasticity and cross-link domains. Hydrophobic carboxylic terminal sequence consists its only 2 crystatin residues. So next slide. For tropoelastins, domains each arise from single exon of ELN gene. They can be categorized as either hydrophobic or cross-linked based on their functionality and amino acid content. Tropoelastin's primary sequence is low in complexity and contains repetitive motif. Furthermore, tropoelastins, hydrophobic and cross-linked domains are arranged in alternative patterns throughout the majority of the molecule, giving rise to both inter and intra-domain level repetition. Tropoelastin sequence are domain arrangement. Tropoelastin is a low complexity protein on both primer and secondary sequence level. It hydrophobic and cross-linked domains consist repetitive motifs that contribute ununique to elastin assembly. The hydrophobic domains contain alphabetic amino acid which proline variation that provide flexibility and ability to assemble to higher order structure. The cross-linked domains are enriched to either list pro which is Kp or lys La which is Ka motif and form cross-link that link growing tropoelastin chain during elastonogenesis. So next. The cross-link domains are defined by lysins which are responsible for the formation of durable bi, tri and tetra functional cross-link within mature elastin. Cross-linking domains can be subdivided to Kp or Ka domains, which denote the amino acid, proline or alanine respectively, that are adjacent to the lysine. Kp domains are found closer towards the N terminus, whereas Ka domains are closer to a C terminus, include the alanine tracks. Unlike hydrophobic domains and cross-link domains in isolation are unable to self-assemble. The amino acid sequence are domain 36 is unique. Furthermore, the sequence confers a positively charged C terminus comprising linearness between positively charged arginines, forming a RK-RK sequence. In addition, the RK-RK sequence domains 36 content of tropoelastin solo 2 cysteines and only disulfide bond. Perturbation of either the compound are greatly reduced. Tropoelastin able to self-assemble into a vitro and interact with the microfiber of elastin fiber, which interact domain 36 require for correctly assembly. That's all from me. For conclusion, I pass to Vera.
Thank you, Siri, for the part you presented. Now we are coming to the last part of our presentation, that is conclusion. Now, our body tissue also loses their elasticity. They can never regain it again. It can be reduced or damaged due to various reasons like stress, hormone, and smoking. It can also be due to aging. As the body and its organ loses elasticity, our body's functioning is also affected. Elastin is also damaged due to exposure to the sunlight. Elastin is one of the proteins that is more long-lasting in our body. This is the end of our presentation. Thank you for your time and everything.